In continuation with the last class, we are discussing today about the DNA polymerase 1. Okay, this is a classical DNA polymerase present in the bacteria. It's a bacterial DNA polymerase. Okay, and it is one of the important polymerase that is used in replication in the bacteria. So its occurrence is bacteria. So the source of this particular DNA polymerase is bacteria. So basically it's a bacterial DNA polymerase and it is involved in the replication process. Okay, DNA polymerase 1 will be having the three important functions. Okay, when we talk about the DNA polymerase 1, it will be having the three important functions, and remember this will uses the magnesium as cofactor. DNA polymerase 1 will uses the magnesium as the cofactor. So and you need to observe one more thing when we are studying the enzymes used in genetic engineering. You should also remember what is the cofactor used by that particular enzyme. Some enzyme doesn't use cofactor, there need not to worry, but wherever a cofactor is mentioned, you should remember properly what is the cofactor for which enzyme. Okay, coming to the natural functions of the enzyme. So functions or activity. Activity or function means like what does it naturally, you should know the difference between the functions. So functions mean naturally what for that particular enzyme is there. Application is a different aspect. So function and application are different. Function means naturally what is the purpose of that particular thing. Application means how we will use that one in for our benefit. Okay, function means what it has been, what work it has been allotted by nature to do. Okay, application means how we will use that particular thing to derive the application for human welfare. That difference you need to remember. Okay, coming to the functions or activity of DNA polymerase 1. So DNA polymerase 1 will be having the majorly three important activities or functions. One is 5' to 3' DNA polymerase activity. 5' to 3' DNA polymerase activity. Polymerase activity. And second one is 5' to 3' exonuclease activity. Okay, third important thing is 3' to 5' exonuclease activity. Okay, so 5' to 3' polymerase activity means it will, what it does, it will, this enzyme, this particular DNA polymerase, what for it is basically designed for? Polymerization. So it will add up the nucleotides to the at the 5' end of the DNA. That is your primer. Okay, 5' to 3' DNA polymerase activity means it will add up the nucleotides to the 5' end. Okay, at the DNA. 5' to, this is the basic activity, that is to get a new copy of the DNA, it need a primer, a template and a primer. So template it will take as a reference, and to the primer it will add up the nucleotides and extend the primer to make the new copy of the DNA. That you just follow the figure which I have given in the last class, you will understand in a better way. Okay, this is the first activity. Second activity is 5' to 3' exonuclease activity. Exonuclease means we have discussed earlier also. Exonuclease means removing the nucleotides from the end. Say this is a strand. Okay, 5' to 3' end. So what this particular enzyme will do is, so this is in the first, before the exonuclease activity will start. Next what happens? So it will become little bit shorter. In the next instance, so little bit shorter it will become, and later it will vanish. So exonuclease means removing the nucleotides from the end. Okay. Okay, removing the nucleotides from the end. Removing the nucleotides from the end means it will remove the nucleotides from the 5' 3' direction. 5 prime to 3 prime. So this is the 5 prime to 3 prime direction, means from this end it will start removing. In this direction it will start removing the nucleotides. So that is known as the exonuclease. 5 prime to 3 prime exonuclease activity. Okay. Third important activity, 3 prime to 5 prime exonuclease activity. So here what we have observed here, is 5 prime to 3 prime exonuclease activity. So 3 prime to 5 prime means, see remember the difference. 5 prime to 3 prime means it is removing from the 3 prime end. 3 prime to 5 prime means it will do in the opposite direction. That means from the 3 prime end it will start removing. Say this is the thing. So here, so from say here, so from this end it will start removing. Okay. So it will start removing in a opposite direction. Not this. Okay, now still it reduces and then reduces further and then vanishes. Okay, from the reverse direction. Here the exonuclease is from 5 prime to 3 prime and here it is in the reverse end. Okay, it is having very much significant feature, this particular thing. That's why it is also known as the proofreading activity. Proofreading activity in the DNA replication. Okay, this 3 prime to 5 prime exonuclease activity is also known as the proofreading activity in the DNA replication step. So what happens is here, so let me explain further. Okay, in the DNA replication what happens, you have a strand like this. Okay, template strand. For basically it will be having a 2 strand. Later this 2 strand will become a single strand and a primer will be there, and our polymerase will be keep on adding the nucleotides to this particular end. Okay, and then when it adds, finally we'll get a new copy of the DNA. Okay, this is what is the function of the DNA polymerase. So proofreading activity means what? Okay, sometimes what happens is when it add up the nucleotides, it doesn't add up properly. Say example, so it will add up, it will follow the base complementarity. Base complementarity means if the G is there in one strand, it will add C. If A is there in one strand, it will add up T, like vice versa. If C is there, it will add up G. If the T is there in the template, it will add up A. Sometimes what happened, there is some mismatch has happened. Okay, to remove that, so whenever we write something, we will again recheck. No, that is known as the proofreading activity. So what does it do is, so this here, so once it completes here, it will come back and rechecking. So it will do the exonucleotide activity from this direction. So that's why it is known as the proofreading activity. It will start checking from the opposite end. From here it will come and check. Is there any mistake here? 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 And it will does the exonuclease activity. So that's why the 3 prime to 5 prime exonuclease activity is also known as the proofreading activity. Proofreading activity. Okay. DNA polymerase 1 is having 3 important activities. One is 5 prime to 3 prime polymerase activity, 3 prime to 5 prime exonuclease activity, and 5 prime to 3 prime exonuclease activity. Okay. Now coming to the applications of enzyme. Applications of DNA polymerase. First one, obviously it is useful in the DNA replication. Okay. Application means it will be used in the synthesis of probes. Synthesis of probes. So this will discuss later when we discuss about the hybridization, probes and hybridization. This time. So synthesis of probes, synthesis of synthesis of seed library, DNA library. This also we'll discuss later. And then in the, so before the advent of PCR technique. Before the advent of tag DNA polymerase, this is used as a basic enzyme in the PCR technique. Okay, this completes your DNA polymerase 1.